Okay, I'm recording my session from here on. In the last part, we have studied the ribose switch, and there are a few more ways, few more methods in which a cell can control its expression of the genes. The next is the very famous, the alternative promoters and alternative internal promoters. Until today, with whatever the understanding we are carrying about our control of gene expression, if you can recall your understanding about the control of gene expression, you know that typically a promoter region is supposed to be present upstream of the transcription start site. The transcription start site is typically represented like this. So this place is supposed to be the promoter region. Now, these are the baby stuff for you now. The promoter region is the place where the transcription initiation complex is assembled. And then it is supported by X, Y, Z, so many things which will define that how much expression of this gene will be executed. So we have gone through all these things a lot. So one more thing you need to know, that in some cases, a one gene can have multiple promoters. How? So this is supposed to be the one of the axon. There is another axon over there, and you can guess in between the axons is called as the introns. Sometimes because of the structure of the genes, because of the sequence of the gene, two different positions on the gene can act as a transcription start site. So this one gene can initiate the transcription from here. It will going to produce a messenger RNA, will be like this, that much long. But the same gene can also start its expression using a transcription start site present here. In this case, the messenger RNA will be like this, will be that much small. Now that gene, if it start the transcription start site from here, if it use this site, that portion will going to act as a promoter. But luckily or unluckily, it also contains another transcription start site within itself because of the sequence of the genes, because of the sequence of the DNA. So for this transcription start site, that portion act as a promoter. This thing will going to act as a promoter for this transcription start site. So now what will happen? Either this promoter is used and this particular messenger RNA will be created, or either this internal, or I must say alternative promoter can be used, and it will going to produce another messenger RNA. Typically, this type of promoter which is present within the gene is called as alternative internal promoter. It's called as internal promoter. And you can guess, if the internal promoter is there, it is the other promoter will exist between the axons, and that will going to produce multiple messenger RNA, a new type of messenger RNA that might be small. It might not produce any protein. It might be truncated. The concept of alternative promoter is that sometimes one transcription start site can be controlled, can be regulated with the help of two different promoters. So this is the one of the promoter which can activate this gene, but this gene can also be activated with the help of another promoter here. So two different promoters can influence the same gene. So that in that case, we can have a different length of messenger RNA might be. We might have a different primary messenger RNA because of the use of different alternative promoters present here. In order to grab that thing perfectly in your mind, you need to understand anything which has a sequence like promoter can act as a promoter. Anything who have a sequence similar to a promoter, that portion can act as a promoter. If we have two such similar sequences present here, 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 and there, and maybe inside the gene. All of these three locations can act as a promoter. It can recruit the RNA pol 2, it can create the transcription initiation complex, and it can trigger and start the synthesis of messenger RNA.
Excuse me, sir.
जी
सर जो आपने ये बताया अल्टरनेटिव प्रमोटर का, अगर ये कोई भी सीक्वेंस हो सकती है, फॉर एग्जांपल इफ दे हैव सिमिलर कोडिंग, तो इसकी मतलब कि डिस्टेंस कितना होना चाहिए फ्रॉम द जीन और द ट्रांसक्रिप्शन फैक्टर या ट्रांसक्रिप्शन साइट कि वो फिर उसके ऊपर एक्ट कर पाएगी, लाइक कहीं भी प्रेजेंट हो तो इट कैन एक्ट ऑन.
Good question. That's a very good question. If any sequence which is similar to the promoter sequence can act as a promoter, then where it should be located? It should be located within the premises of the gene. I mean, I can't tell you the exact distance, but the premises of gene mean where the transcription or RNA pol 2 can proceed with a transcript. It can produce a single transcript out of it. So it should not be very much far away, because it can lead to a messenger RNA which is like extraordinary length, is of no use. So it should be like near in the premises. It's not very much far away. Okay, I can't give you the exact number, but it should be close to the gene. In that case, it can, because if you have a promoter, yes, if you have a promoter over there, but if you don't have a gene, so what will happen? क्या होगा? Guess करो क्या होगा? If you have a promoter but not the gene, the RNA pol 2 will be recruited, but it will not going to do anything else. Just bind and then leave, bind and then leave. Throughout our genome, in whole of our genome, different RNA pol 2 are like present in different locations. अगर हम genome को zoom करके देखें, तो हमें पूरे genome पे जगह जगह पे RNA pol 2 लगा हुआ मिलेगा. And logically, if you can zoom in further, you came to know that these regions जहाँ पर where the RNA pol 2 is located, these are promoter looking stuff. अब RNA pol 2 की आंखें तो है नहीं, they just find the similar looking sequence, binds there. But if there is no gene, if there is no transcription initiation complex, it will not going to produce anything. Okay, and next there exist different RNA binding proteins. They can also regulate the function. The way is very simple. You know, when the messenger RNA is created, it has a 3' UTR, it has a 5' UTR region, and different proteins can attach themselves to the RNA molecule at the 3' UTR region, sometimes at the 5' UTR region, and they can influence the ability of that messenger RNA to go for the translation. And that is how the untranslated region attach with different proteins, and those proteins can block the translation or can push the messenger RNA to go for the translation. That is why some people wonder, and you might wonder as well, that this untranslated region, I'm trying to draw a messenger RNA, and this is the portion which belongs to exons. And you can guess here is the untranslated region, and there is an untranslated region. No amino acid is incorporated from here and from there. All the amino acids came from the exonic region. So people might wonder that what is the use of this rest of the portion of the messenger RNA? They are doing wonderful job, I can tell you. They are helping the messenger RNA to survive. There are certain proteins present here. They are helping the messenger RNA to transport from the nucleus into the cytoplasm, and they are helping the messenger RNA to find the ribosomes, and they are also sometimes blocking the expression of messenger RNA by binding on the untranslated regions. So the regions which are not involved in translation on the messenger RNA, they do have a significant role in controlling the expression of gene. But having said this thing, I wish you understand and grab the statement. Please, Sonani, बहुत important jumla ye. Once the gene is transcribed, a messenger RNA is created. Whatever the regulation comes after that is called as post-transcriptional regulation. So this is the kind of post-transcription regulation. You can regulate the gene before it transcribes. Whatever the things you have studied, cis-regulatory elements, trans-regulatory elements, all those were regulating the expression of gene before it actually transcribed. Whether it will going to transcribe faster or slower, But once it is transcribed, it will become messenger RNA. Still, nature holds its control. Nature holds a way how to control the expression even after the messenger RNA is created. Why? With the help of RNA binding proteins. It can
Sir?
It can be present on the untranslated region and can downregulate the function even after the expression, even after the creation of messenger RNA. जी हाँ.
सर आपने अभी इंट्रॉन्स का फंक्शन बताया है कि ये ट्रांसपोर्ट के लिए होता है कि न्यूक्लियस से साइटोप्लाज्म में.
मैंने नहीं बताया ये.
नहीं मतलब प्रोटीन्स इस पे बाइंड होती हैं जो कि ट्रांसपोर्ट करती हैं न्यूक्लियस को.
इंट्रॉन नहीं है ये, अनट्रांसलेटेड रीजन.
हाँ जी सर, लेकिन स्प्लाइसिंग जो है वो मैसेंजर आरएनए की वो तो न्यूक्लियस के अंदर हो जाती है, तो ये इंट्रोनिक रीजन तो सारा निकल जाता है.
मैंने कहा ही नहीं है, आई एम ट्राइंग टू एक्सप्लेन. मैंने इंट्रोनिक का वर्ड यूज नहीं किया है. दिस इज अनट्रांसलेटेड रीजन. दिस इज नॉट द इंट्रोनिक रीजन.
ओके
okay. Untranslated region means this everything here, from here, it's exons. All the introns are spliced out already. Okay. There is a stretch over there. This stretch is called as untranslated region because ribosomes do not put any amino acid using this portion.
Yes sir.
Ribosomes do not put any amino acid using that last portion as well. So one of the portion is called as 5' untranslated region or 5' UTR. The other is called as 3' UTR or 3' untranslated. It's not intronic region. Okay. These portions can be used to regulate the messenger RNA. How? Certain proteins can bind here and it can block the translation procedure. So even the messenger RNA is there, even then that messenger RNA will not going to produce any proteins because of the presence of those proteins. So nature can still control it. Okay. जो आप कह रही हैं अब हम आप बताते हैं. And that is alternative splicing. This is another way to control the function of gene. It is not actually the control the function of gene. It is like what type of gene we want to express out of a single gene. That is called alternative splicing. So if we have multiple exons, we can use different sets of exons to create different types of messenger RNA out of the same gene. And that job is done with the help of specially designed enzymes which are made up of RNA, mainly made up of RNA, and that is called as spliceosomes. It's a very complicated type of thing. It's a very special thing. It's a very special structure, but they are made up of RNA, and those RNA molecules are specially designed how to clip the introns out of the primary messenger RNA. Remember, the primary messenger RNA contains all the portion, contains introns and exons as well. And then the spliceosome machinery, it clips the introns and release and remove those introns out of the primary messenger RNA, converting it into a perfect messenger RNA. But what type of exons will be there? That will be defined by alternative splicing. G, we have two hands raised. कौन कौन है? एक जरून, जी जरून, और फिर हमजा. शाबाश.
सर वो तब कई पहले कई वो हैंड रेज्ड.
अच्छा. ओके, जी हमजा.
Sir, do we have any example? जो भी आपने messenger RNA की बात कही है कि certain proteins, or do we have any pathway you can show us that they are?
क्या दोबारा बताओ किसकी एग्जांपल लेनी है?
सर जो आपने भी बताया कि मैसेंजर आरएनए जब बन जाते हैं इवन आफ्टर द जीन रेगुलेशंस.
Yeah. Hell of example. You just need to Google it, okay? So I am telling you the things are out there. You can follow the individual specific example. I can do the Google as well for you if you want. This next session I can do the Google for you. But it's very simple. You just need to type what. आपको क्या ढूंढना है? आपको ढूंढना है post transcriptional regulation. Okay, the regulation after the transcription, and you will came up with the options, and one of the options I have explained. Okay, and the alternative splicing is very famous. I am sure you people understand, but you just need to know.
Sir,
जी.
सर अगर वो पोस्ट ट्रांसक्रिप्शनल रेगुलेशन है तो इसको हम प्री ट्रांसक्रिप्शनल रेगुलेशन कहेंगे. ट्रांसक्रिप्शन रेगुलेशन।
नहीं, प्री ट्रांसक्रिप्शनल कैसे हो गई? ट्रांसक्रिप्शन के बाद हुई है।
नहीं मैं सर इसकी बात कर रहा हूँ जो आपने लेफ्ट साइड पे जो पिक्चर ड्रॉ की थी उसमें जो प्रमोटर और जीन के दरमियान जो
हाँ, वो
रीजन
जो
थे
हम
उनकी
इससे
भी
पहले
रेगुलेशन
पढ़े
हो
हैं,
गई थी ना।
जो हम इससे
जी
पहले
जी
पढ़े हैं, बिल्कुल ठीक है। Whatever we have studied earlier, all of those types of regulations are the regulations before the transcription. That actually that is the primary way of regulation. कि हमें ट्रांसक्रिप्शन करनी भी है कि नहीं करनी, whether we want to transcribe or not, or how much we want to transcribe. But some people might wonder कि once the messenger RNA is created, उसके बाद ये it's free to go for the synthesis of protein. The answer is no. It is still not free. Nature can control it even after the transcription with the help of post-transcriptional regulations. And I can tell you, even after that, the protein is created, nature can still control it by using post-translational modifications. Nature can still regulate. I will give you and show you certain examples in the meanwhile as well. Okay. Okay, let's move further. That how much exons can be utilized. Alternative splicing is clear. There's nothing much if you want me to. Okay, all set for that thing. Or I should show you an example. खाली सफा चाहिए. Okay. अगर कोई गड़बड़ है तो I think it's simple. I have explained it. If you have any question, please. आप मुझे कह सकते हैं सर बताएं, मुझे नहीं पता alternative splicing का क्या होता है. If you have any problem, okay. I guess that you understand it.
सर ये पोस्ट ट्रांसक्रिप्शनल रेगुलेशन ये डिफरेंट है ना मॉडिफिकेशन से?
जो the Kadish wants to ask a question because he heard another word which states post transcriptional modifications. It's a word, it's a literal word which means that something is happening after the transcription, like system try to change the things after the transcription. These are called as post-transcription modifications. And why these things are done? If you ask yourself why these things are done to regulate the expression of the genes. And that is why those are also called as post-transcription regulation. It is a way of using a sentence in English. So modifications are done, and those modifications are done for the purpose, and that purpose is called as post-transcription regulation. Hope this answers the question.
Sir.
जी.
Sir, I am little bit confused कि जो भी आपने modification का word बताया, सर modification का मतलब ये नहीं है कि उस पे आप जो पोस्ट ट्रांसक्रिप्शन में मैसेंजर आरएनए बनता है उसके ऊपर आप 5 एंड पे कैप लग जाती है और 3 एंड पे पॉली ए टेल लग जाती है. दैट इज द मॉडिफिकेशन जो कि प्रोटेक्ट करती है.
दिस इज आल्सो मॉडिफिकेशन
एंजाइमल डिग्रेडेशन से.
Sure. This is the modification, of course. But you can see, at first, the messenger RNA which is created, it contains these three exons and the introns. Okay, this is called primary messenger RNA. This messenger RNA came out of the DNA. RNA pol 2 create that long messenger RNA. Okay. After that, what will happen? After that, first thing, 5 prime cap लग जाती है, जैसे हमजा ने बताया. Of course, introns splice out कर जाते हैं. Okay, and these 3 exons joins together, and then we have like this type of messenger RNA with 5 prime and 3 prime untranslated regions. After that क्या होता है? Certain proteins can bind themselves here, certain proteins can attach themselves here. So what are all these things? These are post transcriptional modifications, and these modifications are changing the messenger RNA after the transcription, and some of those modifications are regulating the expression of messenger RNA. Okay, जैसे मैंने बताया, the RNA binding proteins, this one and this one, is post-transcriptional regulation. Splicing, like this one and this one, intron को जो splice किया हमने, post-transcription modification. So these modifications are changing the pre-messenger RNA into a final version of messenger RNA. And then attachment, of different proteins on the messenger RNA untranslated region to control the expression. Okay, shabash, good. Essay, it's wonderful to speak and to get yourself cleared. Okay, shabash. Next as well. I was trying and I have started to explain you guys about humans, the female contains an X chromosome, while the male contains an X and a Y chromosome. And Y chromosome is smaller in size, contains less DNA. So, and we know they are homologous pair of chromosome. What homologous pair of chromosome means that if a gene is present at this locus, the same gene will going to be present here at the on the same locus on the partner chromosome or on the homologous chromosome. Let's name this gene as A. So if this is an A gene which is present here, the A gene will supposed to be present here as well. Yeah. Okay. In case of Y chromosome, the Y chromosome is different from the X chromosome. So it is not always possible, because they are different, that if a gene is present here called B is difficult to draw on the mouse pad, that same gene will not going to be present on the Y chromosome. It is not there. So what that means, that in case of female, there are certain genes which are expressing twice in number because they are present in both the chromosome. If this portion is generating 1 copy of messenger RNA per day, for example, this locus will going to produce 1 copy of messenger RNA per day as well. They will going to produce proteins and execute the function. So we have like 2 copies of the same gene produced within the system. In this case, the B will going to produce a messenger RNA per day, but there is no B is present on the Y chromosome. So the system will receive half dose of that particular gene because of the expression, because of the unavailability of the same gene in the locus of the other partner. So the female, they are supposed to have higher dose or higher expression of certain genes as compared to the male. You can ask me the question, the concept is that, is this type of dosage do not exist on the other chromosomes, autosomes? You can ask me, sir, if there is another chromosome, autosome pair number 1, this is in the males, and this is the autosome pair number 1 in the female. There is a gene here. The same gene will be present here in the females. The same gene will be present here in the male, and the same gene will be present on the other partner in the males as well. They will going to produce 2 doses. The answer is yes. In females. How about in males? In the males, they will going to produce 2 dosage as well. So this is nothing special. It exists in the other chromosomes. But the double dosage opportunities are present in female as well as in the males. But the same situation is not right, is not correct for sex chromosome. In case of sex chromosome, we have double chance or 2 dosage opportunity for a gene in case of female. But the unfortunate male, they don't have the chance, any chance of a double dosage of the gene which is present on the X chromosome, because it might not be present on the Y chromosome. So that is why the chance of having a greater function, the chance of having an enhanced function of those genes is very much high in the female as compared to the male. So it is required that in case of female, those genes or the genes present on the sex chromosome or on the X chromosome in the female, those genes should be down-regulated by some way. Their expression should be decreased by some way. So some of the genes are switched off on this chromosome, and some of the genes randomly should be switched off on this chromosome. And that will result in almost decrease in the overall dose of individual genes. If the gene is expressed on both the partners, this is like homozygous expression. But if the gene is expressed only on the one partner, it is like hemizygous expression. So what if you switch or turn off certain genes on the X chromosome? You never know which genes you are turning off. But whatever the genes you are turning off here, they will not going to express. How about turning off certain other genes on the other member, on the other partner? And that will going to turn off certain genes on that part. So theoretically, it is possible that for many copies, one of the gene is turned off here, but it is expressing here. And similarly, some of the genes which are expressing here might not be expressing on the other partner because of this selective silencing of the genes. Now, how it is possible to silence the gene on these X chromosomes? That's a very important question. हम जो पढ़ रहे हैं, whatever the thing we are studying is that how we can regulate the expression of genes, how nature inactivate those genes present on the X. The first thing is that we should know why there is a need to silence the genes, and now why it is required, how it can be silenced. It can be silenced by touching the nuclear membrane. Remember, the region within the nucleus which is present beneath the nuclear membrane, all that portion which is present beneath the nuclear membrane, it is heterochromatin region. It is because of the presence of certain proteins. I wish you study those proteins, very interesting. These are heterochromatin region. Anything which come close to the nuclear membrane, automatically the genes present there were silenced. So usually that is the main factor which actually turns the genes down on the X chromosome. It's a random event and it's a reversible event as well. जी, next question, हाथ खड़ा किया किसी ने. कुछ और phenomena हैं. You just try to Google those phenomena like ZIC है, XC or cyst genes हैं. That is how they inactivate different portions on the X chromosome. Hand raise किया किसी ने. जी.
Yes sir, मैंने किया था again. सर वो आपसे पूछना था कि जो ट्रिपल एक्स सिंड्रोम है, ट्राइसोमी, तो उसमें इसका क्या रोल है? उसमें
ऑटोमेटिकली यही होएगा ना. The things I can tell you right now. जी जी, आपका क्वेश्चन पूरा हो गया?
दो हैं बेसिकली, अगर आप इजाजत दे तो पूछ लें.
Sure, why not?
यस सर, एक तो ये ट्रिपल एक्स का था कि जो सिंड्रोम है, इसमें ट्राइसोमी जो होता है बेसिकली, इसमें ओवर एक्सप्रेशन होती है. दैट्स व्हाई वी कॉल्ड इट सुपर फीमेल. ठीक है, इनकी हाइट वगैरह ज्यादा होती है. आई डोंट नो द बैकग्राउंड ऑफ द जीन, बट आई थिंक कि उसमें भी ओवर एक्सप्रेशन होती है. मे बी वहाँ पे साइलेंसिंग नहीं होती, सिलेक्टिव साइलेंसिंग नहीं होती. एंड द सेकंड क्वेश्चन इज दैट कि जो आपने अभी बताया था कि ये जो वाई है, दिस इज अ लेस X chromosome basically. It is a form of the less X chromosome. ऐसे ही है सर?
Not exactly ऐसे है, but we can assume. Okay, मैं अब करेक्ट करता हूँ इसको. मैं इसको रीमोडिफाई करता हूँ अपने स्टेटमेंट को.
Okay सर.
पहले आप पूरा जुमला कर ले, फिर मैं बताता हूँ.
नहीं सर ठीक है, सेकंड क्वेश्चन इज नॉट दैट इम्पोर्टेंट, बट फर्स्ट जो था ट्राइसोमी वाला वो एक्सप्लेन कर।
Every question is important. The first thing you need to understand that when we have an extra chromosome, that means we will going to have an extra dosage of a certain gene. Extra dosage, or not always, it does not always mean that it is going to give you more things, because we never know that how one gene is going to execute its function completely. Sometimes one gene can execute a function and we are lucky to know that function, but most of the time they are interconnected with each other. One gene is involved in another gene's function, the other gene is connected with another gene function, they are connected with a cycle, and some are connected with a metabolic pathway. We never know. It's too much complicated interconnections are out there between the different genes. So having more copies of one particular gene, it does not mean that you will going to have like things a lot. That simple means abnormality in function. Things will not going to perform in a normal way, and that's we can conclude, and that leads to the disease. So extra chromosome means extra copies of something which is not required, and that will going to interfere with the function, and that can lead to anything. I mean, it's so difficult to rationalize. But since the people are born with extra chromosome, so that means somehow they, they manage to develop in the womb of the mother with this abnormality. But with the passage of time, when they grow, they will be, we can see more and more phenotypical errors in them. It is difficult for them to live in a normal life because of those errors. But since it's an extra dosage stuff, it's an extra dosage type of situation. So that is why it is not lethal during the development. The people manage to live. Extra dosage give them bad phenotype, give them abnormal phenotype. Okay. The second thing is that the Y chromosome, when I said, actually, in the perspective of evolution, both of them should be same. Like life arises from a single gender, so they are supposed to be the same like the other autosomes. It should be same in both. But the better version of life is to divide life into two genders. It gives more diversity, more chances, and eventually humans. So Y is an answer of a long-term evolutionary workout of nature. It is supposed to be same, but eventually they started like becoming different, and we acquire a lot of things at the same time. So Y contains lots of genes from similar to X, and does not contains many genes which are present on the X as well. And there are some genes which might be there which are not present in the female, specific on the Y chromosome. And I can tell you, during the development, I wish you people study development. That's very wonderful. I always encourage you to study the process of development. At one point of time, you cannot distinguish between the male and the female until unless a very famous gene expressed, that's called as SRY gene, SRY. Do study that gene, okay? The appearance of that gene defines the outcome of that fetus, and that will started showing the difference in male and the female. Before that, everything looks very same, and you will see how it comes from the Y chromosome and it started influencing everything after that. वो जो लोग सोचते हैं ना, wow, it's going to be a male baby or it's going to be a female baby. So that is the time when actually it started happening. Because of I sometimes wonder, and you can study as well, if the SRY gene is silenced, what will happen after? Because it controls so many other things at the same time, so it is quite difficult for the fetus to survive without the expression. Okay, my next question is that I have the question from at what stage does X inactivation occur? The X inactivation should occur when the X chromosome is going to be part of the next generation. Like the zygote, when it receives an X chromosome from the parent side, it receives with this random inactivation of certain genes. Okay. And there are other methods and mechanisms which you can browse. That's good enough for our class. Okay. And then there are maternal mRNA. Apart from this, you should know about the thing. वो याद है आपको? Mitochondrial RNA only belongs to the mom. I have made a video out of it as well. It's very, very wonderful phenomena. You are carrying all the messenger RNA in your, sorry, all the DNA in your mitochondria that belongs to your mom. And it is because of if you know the contribution of individual, like egg is quite big. Actually, it is more big than this in scale. The sperm is very small as compared to the egg. So a lot of sperms, they are striking, they are trying to penetrate into the egg, they are trying to use their enzymes to melt or to create a pore on the walls of the egg with the help of the enzymes, and during that process they eventually able to unload whatever the material that stuff is carrying. Once the wall of the egg is breached, the egg release a protein which sealed it off. That is very important because hundreds of sperms are trying to enter. So now no other sperm is able to penetrate the egg. It brings the genetic material. The egg also contains the genetic material as well. The egg also contains, apart from the genetic, this is the nucleus of the egg, and this is the nucleus of the sperm. The egg also contains mitochondria, so whatever the energy which is required to execute the functions, enzymes, messenger RNA, they are present here. And we have studied a very wonderful thing during our classroom, if you can remember, how the first difference occurred after the fertilization. So the DNA present in that mitochondria is contributed by the egg. Though two nuclei will going to merge, and that will going to create a zygote. And remember, when the zygote is created, it contains a mitochondria, and that mitochondria came from the mom. Because the sperm does not bring any mitochondria. Sperm के पास एक life होती है, small short lifespan होता है, and that's all. Now, after that, we know about the cell divisions, and you should know one more important thing. The cell division, the cells are not growing after that. Zygote does not grow after that. Kyun? Because all the energy which is required to execute the functions, the initial functions, all that energy is contributed by the egg. And you can see the similar phenomena is there in other animals as well. The egg is not only contributing the genetic material, but egg is also contributing the energy and the resources which is required to build the new structure or new baby. If all the energy is present within the egg, if all the energy required to build an organism is present in the egg, what will happen? In that case, complete baby is created within the shell, within the egg. And that happens in case of chicken and in case of birds and reptiles, because their egg contains all the necessary requirements, all the necessary food and all these things. Is it wonderful? I mean, put all the things there within the egg, and that is fine to raise a baby. Why, taking so much pain, labor pain and all these things? But evolution does not consider that thing as a best possible way. What is the best possible way? The best possible way is the one the humans are doing, keeping the baby in the womb. Ahsani takweem hai na? So this is the best way. So you should ponder and think how this is the best way, because if you put everything in the egg, I mean, the eggs are supposed to be present at somewhere they are not very much safe and blah blah things. But in case of humans, the egg is contributing less energy. Okay, there is a species between humans and the birds. Their egg does not contain enough food to create a complete baby. Their egg do contains enough food, but not enough food to create the complete baby. You know, example बताऊं? Kangaroos. They create the egg, and the egg contains the food, but the egg was not shed from the body. The egg stays within the body during all that time, but does not make any connection with the mom. So whatever the food present, because they failed to develop a proper placenta, it was something in between. Before that, आपने सुना होगा duckbill platypus. They produce eggs and lay the eggs. And then kangaroos, they produce eggs. They try to establish a connection with the mom, but that was not a very strong connection. So whatever the food which is present in the egg is the only source for the baby to grow. So what happens? The kangaroo baby was born like this one. कभी आप YouTube पे देखना इतने से babies, few inches, weak, almost dead, because not enough food. So that is why evolution gave them the pouch. They put the babies in the pouch. The babies stay there in the safety of the pouch and then feed themselves over there, and then they grow. So now the next level is that you have the egg and you have a wonderful way of supplying food. And what is the most wonderful and reliable way of supplying the food to the egg? Is the mom. How to establish a connection with the mom. So the slide which I was trying to show you, the thing which I was trying to draw actually, that the egg does not contain enough food. So the zygote is required to create a connection with the mom, establish a placenta. But before that time, now imagine, before that time, there was no external supply of food. So the zygote is that big, it will divide, it will become smaller, it will divide further, it will become smaller. So you will see in the movies or in the animations, if you see the earlier cell divisions like this, it will divide into two, that it will divide into three. The overall size will stay same, and the cells become smaller and smaller and smaller. They are not growing, they're getting smaller in size because there is no supply of food until unless they roll over within the walls of the ovary and then eventually find a spot and then attach themselves and develop a connection with the mom and start receiving food and nutrition, and then they starting growing. Okay, so this is just an extra stuff for you guys. Okay, just to enjoy the wonders of developmental biology.